and classification. We have replication of viruses, viral pathogenesis, diverse or different viral disease and epidemiology, means of viral infection diagnosis. How do we diagnose viruses? Which tools do we use, and so on for each particular conditions. So this is an overview of the unit as prepared by Prof. Muguni, and probably he will be joining for some seminars if deemed relevant, but also if he has time for that. So we still need to find how he can be able to contribute to the unit. And as the module leader, I think he will be able to give some important lessons, important seminars, 1-2 hours. We find out on how this can be done remotely, of course. Any question on this overview? Any question on the overview? I think not. So again, all slides are from Prof. Muvunyi, and you will see that they are very well elaborated, and we can discuss each concept and have an overview of most prepared material. So viral definition and history. Do you see the screen? Do you see the screen?
Yes.
Yes. So what are viruses? So I think you already know this, yeah? But of course we need to really elaborate on this, as sometimes in your previous learning you didn't go too much in what are viruses in sense of elaboration, know the component of viruses, know the difference between viruses and other potential pathogens. So viruses are distinct from living organisms. So they are particles that can have property, living property in the cell, but really not viable outside the cell. So inert outside the cell. So the simple definition of viruses is they are submicroscopic, they are very small, and obligate intracellular parasites. They don't have life outside of the cell. So they should be in the cell to be able to reproduce, to be able to make some important function of living organism. Otherwise, they are completely inert. So viruses are smaller than bacteria. They range in size between 300. Maybe I do this.
Sorry, lecture, I cannot hear you.
Oh, sorry. Hello? Hello?
Hello? Hello?
Yeah, I think I was muted. Sorry. So
Yes.
I wanted to tell
Yes.
you or to show you how small is viruses in terms of size. So this is a red blood cell. Maybe I have this is a red blood cell. Do you see?
Yes, we see.
Yeah, a size of red blood cells compared to a bacterium, a E. coli bacterium. You see that the bacterium are quite small, but not as small as that. And if you magnify this bacterium to 3,000 times, so you are going to get something like this compared to something of this size. This is a phage, bacteriophage, T4, and this is a virus size. And if we see in here, the virus like poliovirus, the cause of polio, is almost the size of the ribosomal structure of bacteria. So 25 nanometer, 30 nanometer are not quite different. Some bacteriophages are even smaller than this structure. So this is just to show you that the size of viruses are such very small. And this is one of the largest virus. Smallpox viruses. Poxvirus is one of the largest and can only measure 200 nanometer to 300 nanometer. So it's the biggest and have this kind of size compared to the bacterium. Do you see? So this is just to show you an idea of what is a virus. If you also see this is one of the plant virus, tobacco mosaic virus, which is very small in size, although longer. It is longer, but very small in size, 15 nanometer only, 300 nanometer. So this is just to give you an idea of how viruses can be represented compared to the human cell, like red blood cell, bacteria, and a virus. So otherwise, we continue. So viruses are such small, as we said, and contain only one type of nucleic acid. They either have DNA or RNA, but never both. This is a very important criteria. So while other living organisms can have always have these molecules at the same time, DNA and RNA, in viruses, the viruses structure have only one type, either DNA or RNA, but never both. Viruses consist of nucleic acid surrounded by a protein coat. Some viruses have additional lipoprotein envelope, which is mostly derived from the infected cell we see. And some other viruses lack such an envelope. They don't have that, and they are called naked viruses. And viruses lack or do not have organelles. No, they don't have ribosomes, they don't have mitochondria, they don't have endothelial reticula, they don't have most of structure or organelles of the living cells. So how do they live then? They have to use the host, and that is why they are always intracellular parasites. So they can't do their own life. They have to depend to the other organisms to survive or to replicate or to do their function. So as we said, they are obligate parasites. Why? Because they don't have organelles, they don't have nothing. So they replicate only inside living cells, and replication is by synthesis of the viral protein. So the viruses replicate and by replicating their genetic material, but also synthesis of protein, mostly or always using the host cell machinery like ribosomes and so on. So they don't multiply. In media, you cannot grow the virus. of course, there is a way of growing the virus in vitro. So it requires what is called cell culture. So you have to really have the host cell to always ensure viral replication or growth. They don't do binary fission, as we have seen for bacteria. They don't do that. And they are absolutely dependent on the cell, as we said, the host cell, for their function. Outside the cell, they are inert. They don't have any living property outside the cell. So as a comparative measure, so viruses cannot grow on the media. To grow, you need really to supply some line of cell that we host these viruses. They don't do binary fission, different from all other bacteria. They don't have both RNA and DNA. They only have one. They don't have ribosome or other organelles. Sometimes, as you can see, they can pack accidentally the ribosome from the host cell, but this ribosome have no role in viral protein synthesis. They don't pack them for synthesis. It's just taking them out of the cell as an accident when assembling. Whether they have muramic acid, no, they don't. And sensitivity to sensitivity to antibiotics, they don't. Whatever concentration of antibiotics, they are not susceptible to antibiotics. So as a brief, they are submicroscopic entity consisting of a single nucleic acid surrounded by a protein coat and capable of replication only within the living cells of bacteria, animals, or plants. Not only bacteria, protozoa as well, fungi, or even helminths. All kind of living cells have their own specific viral families that infect the living cells. So can we judge whether they are alive or not? Inside the host, viruses seems to be viable, seems to execute most of the living function, including replication, multiplication. However, outside the cell, it means the viruses are mere complex assemblage of metabolically inert chemicals, so they don't do any function. While within the cell, this is a cell. This is a cell, and you can see that the virus is replicate. It does, while outside the cell, it does not do anything. It is just a structure, a complex structure of metabolically inert chemicals inside the virus. So some related entities, we say viroids are very small than the nucleic acid of complete viruses, and mostly they are circular RNA molecules with a rod-like secondary structure. They don't have capsid. We see what is capsid. This is the protein coat in which the nucleic acid is enveloped, so it protects them. So they don't have this. So they are they don't have capsid, and their replication is more or less similar or like viruses, and they are also obligate intracellular parasites and are more causative agent of plant disease, not human. They are no viroids in human. Virusoids are also satellite viroid-like molecule. Some are a bit larger, and are dependent on the presence of virus replication for multiplication. So they goes with viruses. So these are they don't have these protein codes, and they are packaged always into virus capsid as passenger. They don't have their own way of infecting other cells, except going with a specific virus. So we see that some viruses goes with this kind of nucleic acid, which also have the property of replicating and causing disturb to the host cell. Yeah, apart from nucleic acid, as we've seen, virusoid, we have also prions, which are more protein. They are infectious agents consisting of single type of protein molecules. These are encoded by gene, also found in normal uninfected cells. So they can be in infected cells, while they mostly and they are associated with pathology like Creutzfeldt-Jakob disease and Kuru disease in human. So these are infectious protein. You can read more on this, but they are also related to viruses. So brief history on virology. The viruses have been kind of known since a long time ago. You can see that 3,700 before Jesus Christ, first written record of virus infection consists of the Egyptian hieroglyph, ancient Egyptian, typical clinical symptom of paralytic poliomyelitis. You can see they had already knowledge of this kind of symptom, and these have been found in their museum, and some even rest like Pharaoh Ramses was kind of died with smallpox. They are kind of lesions that shows that he died from smallpox. And these mummies shows that the viruses was that with human such long. So since long, you can see it's a long time, and killing people, like even the kings. So it was smallpox was an endemic in China. In response to the practice of variolation was developed. That variolation is just inhalation of dried crust from smallpox lesion like snuff. So it gives kind of immunization, kind of vaccine of today. So recognizing that survivor of vaccine outbreak were protected from subsequent infection. This is already the start knowledge on vaccine. So they already see that those people who survived the previous or the the past outbreaks were protected from subsequent infection in so meaning that by infection, people developed immunity that protected them for long in their life. And variolation, which involved inhalation of dried crust from smallpox lesion like snuff, was already in practice in China in those days. So this is the initial idea on vaccination. So only after those long, that is where Edward Jenner used cowpox infected material obtained from the hand of the patient, the young patient, a milkmaid from his home village of this, used this to successfully vaccine 8 years old boy. So this was like the initial step of learning how to challenge the smallpox using antigen of cowpox, infected material of cowpox, challenge the boy by deliberately inoculating him with material from a real case of smallpox. He did not become infected. So by this effect, Edward Jenner already see that the patient or the individual person can easily be protected by injecting the material or by using the cowpox antigen to protect new patients. And this was the start of vaccine development by this man, Edward Jenner. So all these were smallpox was one of the terrifying disease, very contagious and killing quickly the people. So having this effect of protecting people was a very successful or very big achievement by this UK Edward Jenner. And at that time, they didn't know really what was the cause of this disease. They didn't know what is really causing this disease. However, it was not until Koch, Robert Koch and Louis Pasteur, who really clarified on the causative agent of infectious disease, including bacteria as well as viruses. They jointly proposed germ theory of disease in this 1880. So that is when people started knowing, or that is the genesis of microbiology, with Robert Koch and Louis Pasteur clarifying on the cause of infectious disease. And Koch defined famous criteria now called Koch's postulate, same as we have seen, which are still generally regarded as the proof that an infectious agent is responsible for a specific disease. However, as we say, these have limitation. If you can't grow the virus in the laboratory, then Koch's postulate will not be useful. So now there is molecular additional where by diagnosis or by finding nucleic acid genetic material of the same infectious disease in the infected individual, you can confer the cause of that disease. The agent must be present in every case of disease. We have seen this. The agent must be isolated from the host and grown in vitro. This is a bit challenge for virus. To grow viruses, it was a bit difficult because they require cell to be grown on, and that is only when the viruses then were explained, the concept. The disease must be produced whenever a pure culture of the agent is inoculated into a health susceptible host. And the same agent must be recovered once again from the experimentally infected host on the media. So by these four postulates, these four element concept, of course, most bacteria pathogen can be proved the cause of linked to their disease. But of course, as you know, the viruses will not grow in vitro, and you cannot experimentally regrow this once. So Louis Pasteur worked extensively on rabies, which he identified as being caused by viruses. And viruses in Latin, that is poison. He did not discriminate between bacteria and other agents of disease. So although he saw, or he has this great demonstration that the rabies is caused by viruses, but he did not differentiate or discriminate the viruses from bacteria. Why? Because it was not easy to to recover these viruses, to have these viruses. So Dmitry Ivanovsky described the first filterable infectious agent, which is tobacco, mosaic viruses, and smaller than unknown bacteria. That is like if you use a very thin filter, you can be able to remove all other bacteria, but the tobacco mosaic that you have seen, 15 nanometer of, so was kind of not filterable. So you cannot filter this agent. And he was the first person to discriminate viruses and other infectious agents, although he was not fully aware of the significance of this finding. He didn't know what it really means. And that is where virology starts, with this filterable infectious agent. You see that you can use even the filtered material, but you still have infectious agents. And that is the basis of virology, the beginning of virology. And this man, Martinez, extended the work of Ivanovsky and formed the clear concept of viruses, contagium vivum fluidum, soluble living germ. So meaning that they can't really be separated from the fluid. They can't be separated from the liquid. Why? Because the filter that they were using were always larger to let the viral structure get inside the filtered material, and SARS was always infectious for subsequent individuals. And was the person who developed the concept of viruses as distinct entity. So the other previous were like, no, we are not sure this is also kind of bacteria or not. So only this man have seen that these are the distinct entity compared to bacteria and other infectious agents. So these two men also show that the foot and mouth disease is caused by such filterable agents. So they demonstrated that by even filtering, you can still have the virus in your filtered material and can still be infectious. And they are the first people to show that the viruses could infect animals as well as plants. So the previous analysis was mostly based on plants, tobacco, but with these two people, they also show that even within animals, there is such viruses or filterable agent that can infect human cells. Also, in 900, that is when Walter Reed confirmed the yellow fever is spread by mosquito, and also viruses could be spread by insect vectors such as mosquito. So it was the first time that it was linked with the mosquito, although this man did not know the nature of the yellow fever agent. What was it? He and his co-worker were the first to show that the virus could be spread by insects such as mosquito, but they did not link the virus with the cause of disease. They only see the mosquito bite with mosquito, but they didn't link this with the disease. So the polio was also shown or proved in 908 by this Carl, and also demonstrated that it's also caused by the virus. And he won the Nobel Prize because of that very important discovery. And they are the first to prove that virus could infect human as well as animals. Not only animals, but also human beings. Yes. So this man, Francis, was also the first to demonstrate that some cancer are typically linked with the viruses. We see more of this throughout the lecture, and he demonstrated that Rous sarcoma virus, which is a chicken cancer cause of virus, he demonstrated that it is really caused by a virus. And he was the first to show that a virus can cause cancer. And he won a Nobel Prize because of that. So also Frederick showed that virus can infect bacteria, not only human or animals or plants, but also bacteria. And these were classified by Felix in a class of bacteriophage. That is, we have also mycophage. We have, so this is like not only limited to human. So the virus can infect a wide range of living cells, not necessarily human, not necessarily animals or plants. They can also infect bacteria, parasites, fungal, and yeah. So they are more, each virus or each type of living cell have its kind of specific viral agent that can infect them. So only late only recent, this is not too far. That is when human influenza virus was isolated. Although it was transferred from animal to human long time ago and causing problem. If you can know how many people were killed by influenza, like in Spanish flu, where millions of people died from influenza, we still have some outbreak of influenza. Virus and attenuated virus can be vaccine, and show that live attenuated vaccine against yellow fever developed this, and it's still in use today. Not only in the past, but also today, this virus is still in use, or this vaccine is still in use as attenuated vaccine against yellow fever. And this vaccine can stay longer, as we only do up to 10 years. And it saved a million of lives. And sets, of course, model for the production of many subsequent vaccines. If you see the vaccine polio and other vaccine which use live attenuated vaccine. So this comes from this man is a brilliant idea to attenuate the virus and get out with a less virulent strain which can induce immune response and protect the Emory Ellis and Max established the concept of the one-step virus growth cycle, essential to the understanding of virus replication. So by these two people, we had to know how does the viruses replicate. One-step virus growth cycle means from the infection to the or infection from the cell infection to the release of the viruses from the cell to infect other cell. That is a one-step virus growth cycle, showing that the entry to the exit, the entry from outside the cell to the cell, then to exit from the cell. And that was by these two very important discovery. And this work was the base to understand of virus replication, as we say, and do not grow, but are instead assembled from preformed components. So we see that viruses do not grow as you have seen for bacteria, where the bacteria does binary fission. The viruses will not do that. The synthesis of each component is separate, and then after synthesis of components, there will be assembly. So each component will come to join and make the virus, synthesize the virus. But it doesn't work like bacteria, where you have one virus which divides into two. No. A virus will synthesize most of, or from the nucleic acid, it will encode most of the structure components, including proteins, essential protein for the core and the protein codes, and we make this component as separate within the cytoplasm of the cell. At one point, this will be assembled into many virion. In this assembly, we make new viruses, which is quite different from other or the living organism, which does binary fission or mitosis. So this man used an electron microscope to take the first picture of virus particle. So that is when it was then understood, understanding the virus structure. How does it looks like? By visualization was an important advance in understanding the component of the virus, the structure of the virus. And this, as recent as 1949, that is when the virus could grow in cell culture, in tissue culture, by this gentleman, John Ender. Otherwise, it was known on how to grow or to have this virus in vitro. Only this man or these two men were able to grow poliovirus in vitro using human tissue cell culture. And from there, most development comes in, and viruses isolation in tissue culture started from then. In most viruses now, we have this kind of isolating virus for diagnostic purpose, for vaccine production. So it was a very huge advance to have this in culture. This man also proposed the prion, which is more protein, that it's really a slow virus, which at the end did not really retain, show that the course of the kuru is similar to that of scrappy, can be transferred to chimpanzee, and that the agent responsible is an atypical virus, which is not really atypical virus, rather a protein, an infectious protein. So this man also discovered hepatitis B virus, and he won a Nobel Prize because of that. And developed a vaccine. We now have vaccine. And of course, as you know, hepatitis B virus is cause of liver cancer. So it was the first vaccine against cancer because of the strong association of this virus with liver cancer. So the man who discovered the virus also developed the vaccine. And he was living up to recent 2011. Most of you, I think you were there. Viroid, we have seen that viroid are also agents close to virus, but they don't have protein capsid or the package. So these are the nucleic acid, very small nucleic acid, as we have seen, which do not have this coat, capsid coat. So they are infectious, consist of low molecular weight RNA that contain no protein capsid, and were responsible for plant diseases, mostly plant diseases, not human. And David Baltimore and Howard, they co-discovered or independently discovered the reverse transcriptase in retroviruses. I don't know if you know the central dogma of molecular biology. We only say that the information flow from DNA, RNA, protein. So with discovery of this reverse transcriptase in retroviruses, it shows that this central dogma is not retained. It was refuted, and it was possible that RNA can go back to DNA. That is the case of retroviruses, then protein. So it's really a big view on this, and was more discovered because of these retroviruses, the reverse transcriptase, which is HIV class of viruses. So the genetic information flow from RNA to DNA, which was not the known. It was like DNA, RNA, protein. Now with retroviruses, we know that with reverse transcriptase, which is natural, we have RNA, DNA, RNA, protein, which is no part of the central dogma of molecular biology. So, and these people also show that within viruses, there is also some non-coding sequence, the intron. They also showed the genes splicing in adenoviruses. By variating this splicing, the single gene can be able to produce diverse protein. We see that. And this is a very important mechanism by which viruses manage to produce its components with a very small genome. So in 1979, that is when the smallpox was officially declared to be eradicated by the World Health Organization. As the vaccine was such very powerful globally, then it was kind of very successful program, and the smallpox was eliminated. It was very painful and quick killer of the individuals or the infected person. The last natural occurring case of smallpox was seen in Somalia in this year, and this was the first microbial disease ever to be completely eliminated. So even if we have a vaccine for many other bacteria, we still have challenges, or viruses, we still have a lot of challenges in such a way none other pathogens have been eradicated, either polio or so. Saying eradicated globally means even the program of vaccinating is can be shut down. So we still have sporadic case of polio in many countries, sometimes outbreak. So no any other infectious or microbial disease ever have been eradicated. Although we have vaccine, we have treatment, we still have this with us. So this person also showed that the T-cell leukemia is linked to HTLV, human T-cell leukemia virus. Mm-hmm. And this was the first certain human cancer virus to be identified. Although many other cases were seen, but this was the first to demonstrate that this virus is linked with the cancer, and many after were first described were shown to be linked with cancer. We see the list. And this man also showed that prime infectious protein are not really the virus, as we said, although they are related to viruses, as they are also transmissible. They can really increase, and this was the most significant advance in understanding of what were previously called slow virus, which was not really virus. Transmissible spongiform encephalopathy, like this prime protein are also transmissible and can cause really fatal neurodegenerative disease. As you may know, Luc Montagnier, also Nobel Prize 2008, and Gallo, they co-discovered human immunodeficiency viruses in 1983. The causative agent of AIDS, although the description of this virus was, or AIDS was earlier, but they are the one to demonstrate the virus linked to this syndrome. In only 2-3 years since the start of the AIDS epidemic, the agent was identified. So it was a really big effort to really show that human immunodeficiency virus, HIV, was the cause of AIDS, and went quite quick. 2 to 3 years is such very fast, since the syndrome was known in the US. And hepatitis C was also discovered in 1989, and it was non-A, non-B. And although B, A were known, there was some cases of hepatitis where A and B were not identified. And this was the first infectious agent to be identified by molecular cloning of the genome. In most cases, other methods were being used, traditional methods, including viral culture, including electron microscopy, but this was the first to be really identified by molecular approach. So I think we can end with the history. You can read all these. You can have even more additional reading on the history of virology and have more concepts on how the viruses were discovered, development, how people knows how to find out the link between different viruses. So once I share the slide, also in the shared microbiology books, there is some history you can read through and so on. Any questions so far? Do you follow? Can you hear me?
Yes.
Yes.
I can hear you.
So any questions so far? No question? So if no question, we can initiate a new So let's see the structure and how do we classify viruses in general. So as definition, I think you have gone through this. The complete or the virion is the complete infectious virus particle. So we can say it's the complete infectious virus particle. A capsid is the protein coat as surrounding the nucleic acid. Maybe best to go with this. So we have a viral particle, virion, and the viral particle has the nucleic acid inside. Beside the nucleic acid, it have this coat, the coat here, which is which we said this is the capsid, and is made of units, and these units are called capsomere. So they are capsomeres. And some viruses have an envelope, a structure that have this in the nucleocapsid, and some others do not have this envelope, and they are called NECT, while these are called enveloped viruses. So we have nucleocapsid, which is nucleic acid plus the capsid. It may be enveloped. That means it contains protein enclosed by the. So it's mostly from the cell host, some components, some others from the virus. And structure units are the smallest equivalent building unit of the capsid, which we said are capsomeres. Capsomeres are the smallest unit that make the protein coat, the capsid. The structure units are capsomeres. As we said, it either have DNA or RNA surrounded by protein coats called capsid, and this is the basic or the all viruses structure. So either RNA or DNA, it really have to be enclosed in a protein coat that protect this virus from the environmental enzyme from other. So otherwise, the DNA, RNA can be easily degraded by. So the capsid is composed of small structure units that we said capsomeres, and the capsid protect nucleic acid from inactivation by the out physical condition. So when the capsid is removed, then the virus can no longer be infectious because RNA-dependent RNA polymerase, which is a new terminology to you. Normally, it is known that RNA polymerase is DNA-dependent. But for viruses, we have also RNA-dependent RNA polymerase, means RNA replicase, to allow this negative polarity material, genetic material, to replicate. So it goes with this key enzyme, as this enzyme cannot be found in host cell. In host cell, there is always DNA-dependent RNA polymerase, but not RNA-dependent DNA polymerase. So sometimes, or in most cases, or all cases of single-stranded RNA viruses with negative polarity, we always carry this essential enzyme, which we allow them to replicate. Any question on this? No question? Is it clear for everybody? Birumvikana.
Question.
Yes.
Yeah, I don't understand that concept, negative polarity. Viruses with negative polarity.
Yeah, that is what I said. We reach the concept of negative and positive polarity. Normally, we refer to negative polarity, means this RNA cannot are normally complementary to the sense. do you know sense antisense? Or let's say if you want to synthesize protein, so you go you do transcription for RNA viruses. Some viruses or most viruses, RNA viruses, we not go back to DNA process. They will either automatically start encoding for protein, because this is RNA. We say positive polarity when RNA can immediately start encoding for protein. So it's equivalent to messenger RNA. Ukuvuga ngo iyo RNA dufite muri viruses iba ishobora gukoreshwa as template for protein synthesis, kubera ko automatically isa na messenger RNA cyangwa ariyo messenger RNA yiyo virus. So that is positive polarity. While negative polarity means this is complementary. The DNA, the RNA enclosed in the nucleocapsid or in the capsid is in antisense. So it is a complementary to the RNA that is needed to synthesize protein. Urabyumva? No kuvuga hano negative positive polarity is referred to the transcription, to the translation. So translation. So when the RNA can be automatically used as template for protein synthesis, that is positive polarity. And when this require first to synthesize another complementary strand to this RNA, that is the need then of RNA replicase, that is the need of RNA-dependent RNA polymerase. Then this one will be named negative polarity. So it will require this enzyme to first replicate or synthesize the positive sense or the sense, and then from the sense, the protein can be synthesized from that strand. So with negative strand, you cannot immediately import protein. With positive strand or positive polarity, you can automatically start, or it can start protein synthesis. Is that clear to you?
Yes, clear.
So it means positive polarity, negative polarity is referred to the synthesis of protein. Positive is direct, negative means it will not be direct. It will require a synthesis of a second strand, then to the template of protein. We see this in next slides. So as we said, so this is an envelope, outer envelope of viruses, and we have the nucleocapsid, where we have the nucleic acid, and the entire capsid of the viruses. Some viruses have this, some others are just the nucleocapsid. So meaning that they only have the capsid and nucleic acid. This is an example of one of the virus, adenovirus, and viruses have shape. Some viruses have this kind of polyhedral viruses in which the capsid have many angles, and the nucleic acid is just enclosed inside nucleic acid is just enclosed inside here. They may be classified into several morphological types on the basis of their capsid structure, as revealed by electron microscope in the what is called X-ray crystallography. That is the mean of representing the structure of viewed on electron microscope. And we have like two categories. One is those with helix capsid, and those with polyhedral symmetry. They have too many heads or angles, and they can be in 2-fold, 3-fold, 5-fold, depending on the nature of the virus. With helix symmetry, in which the protein subunit and the nucleic acid are arranged in helix. You can see that the shape of nucleic acid will always follow the shape of the capsid. If it is helix, then the nucleic acid will be in helix, while in polyhedral or icosahedral symmetry, the virus nucleic acid also take the nature of this structure. So this are the most important for many viruses, but we also have a complex symmetry where it is not really as helix or icosahedral, but have a symmetry which is combining both or different from either. That is the case of pox virus that we see. Normally, larger viruses often have a complex architecture. So the larger viruses have such complex architecture. That is the case of pox virus, which is the largest. So they can consist of both helical and isometric, as we just said. And the material, nucleic acid material can just be inside the isometric symmetry. And small viruses have mostly they are in are more resistant, and in most cases are not enveloped, and we see that they are more resistant than those larger complex, like herpes, retrovirus, which are larger but also have this envelope, which makes them to be weak or susceptible to easy destroyed, either by dry, by heat, by all other environmental factors. So, as matter of, you can see that this phage, this bacteriophage, a T even bacteriophage, has both helix and the isometric, which is then complex phage, you can see, and to infect mostly bacteria. So basically, the virus structure in simple words, we either have DNA or RNA enclosed in capsid protein, a protective structure, and capsid protein or capsid together with the nucleic acid is called nucleocapsid. And this nucleocapsid have everything that's required by these viruses to replicate, to infect the cell, and these are called next capsid virus. Those are the non-enveloped, or they don't have this lipid membrane glycoprotein. When the nucleocapsid have outside, its outside sheet have lipid membrane glycoprotein, then this is enveloped virus. and you have these two categories. And we see that the property of these two category of viruses is different, as viruses which have envelope, as I mentioned, they are more easily destroyable, while negative capsid viruses cannot be easily destroyable because they are such resistant capsid so it is and while this envelope or lipid membrane glycoprotein can be easily affected by dry heat and so on. So can we have a break of 15 minutes and continue then 10:50? Can we break for 15 minutes? Those with question, please hold. We can have a round of question when come back. So we resume at 10:50. Is that okay for everybody? So we break for 15 minutes and come back. See you. What about a colleague from GACO? Are you there?
Yeah, yes, we are here.
Hello? GACO, you are there, Elvis?
Yeah.
Hello. I hope you are all back. Are you there? Can we resume? Hello?
We are here.
Yes. So are you all back? Can we resume?
Yes.
So as basic structure that we already see is that there is a nucleic acid, either DNA or RNA, one of these, and the coats, the protein coats that we named capsid, and both together, nucleic acid plus capsid are called nucleocapsid. And for some viruses, that is all. That is the type of NECT viruses or NECT capsid viruses. Then there is some other bacteria that have, viruses that have on top of nucleocapsids have a membrane, an envelope made of lipids membrane, basically from the host cell, and glycoprotein sometimes, and these are always having this, and they are named or labeled enveloped viruses. These are two important categories based on the structure of viruses. So in diagram, we have the symmetry, which may be icosahedral or helical or helix. So we have these two, and this can be either NECT, that means there is no envelope outside, or it can have this envelope outside, and this is the case of enveloped viruses. So with enveloped viruses, we can see that you have a matrix inside, you have a lipid outer, lipid membrane, and we have a glycoprotein. We see how these viruses get this structure, normally with budding from the host cell, budding like the replication of or multiplication of some of organisms like yeast. The viruses also bud from, or they do budding from the host cell, and by then they carry or they acquire this outside structure. We see all this. And this is an example of icosahedral NECT capsid viruses like adenoviruses, which is seen here. it is necked, and with electron micrograph, you can see that it's really a compact structure, a capsid, nucleocapsid. And this is a representation of crystallographic model of foot and mouth disease virus of animals. So you can see how the necked virus with icosahedral capsid looks like. So helix, also we have this example of tobacco mosaic viruses, which is really long but very thin, very small. And this is the model representation of how the RNA and protein are within these viruses. It is also necked capsid viruses. So we have icosahedral enveloped viruses like herpes simplex viruses, which have this envelope, and we have nucleocapsid inside the envelope, and these is an enveloped viruses with icosahedral structure inside here, and icosahedral is counted in the nucleocapsid or the capsid structure. So it doesn't count for the envelope, it counts for the nucleocapsid. And this is the cryo model, the cryo electron microscope model of this virus, herpes simplex virus, how you can present this virus with then we have helico-enveloped viruses, helix-enveloped viruses, like influenza A viruses, paramyxoviruses. Paramyxoviruses is a group of different viruses, includes the cause of measles, mumps, all those, or parainfluenza are in paramyxoviruses, and it looks like this in the electron on the electron microscopy. So you can see how the inside it is helix, and the envelope is outside helix, inside outside envelope. So by having the envelope, the viruses have some properties that allow these viruses to be different from the next viruses. Envelope is sensitive to drying, so it is sensitive to drying, so it can quickly be inactivated in case of drying. So they are very sensitive to heat. Even a small temperature can inactivate enveloped viruses. They are sensitive to detergent, as you have seen. The envelope is made of lipids, and detergent will quickly degrade or hydrolyze the lipids and make this virus inactive. So with detergent, you are able to inactivate all enveloped viruses. They are also sensitive to acid. Such consequences must stay weight during transmission. So it means when dry, the viruses just lose its ability to infect the host cell, because without this envelope, then the nucleocapsid is not recognizable by the host cell, cannot get in. Transmission in large droplet and secretion, so it have really to go with these droplets, so it have to to be in wet condition, cannot survive in gastrointestinal tract. Why? Because there is acid. They will immediately be inactivated. They can't do anything. Do not need to kill cell in order to spread, because these viruses need part of this cell, which is the envelope. The envelope made of lipids is unique and have to be from the cell. And if the cell is killed, then most of viruses inside the cell, we then lose this ability to transmit by losing the acquisition, by losing the property of acquiring the envelope. So by killing the cell, then all viruses being manufactured inside, we lose the property to infect, because they don't have this envelope, which have to be acquired during release. We see that. So this may require antibody response, and can be sufficient to clear, to neutralize the infection. Sorry. So we have do not need to kill cell in order to spread. And these viruses mostly will require the immune or antibody and cellular immunity, because once the virus is inside the cell, and as we said, if this cell is killed while the virus is still inside, this will lead to inactivation of these viruses inside. Because of that, they need to acquire. Sorry, a question? Iranzi. A question?
No question.
What?
No question.
Okay. So if killed before the release of this virus, then it will not be, the virus will not be active. So on the other side, on the other hand, next capsid viruses are resistant to drying. They don't really get inactivated by dry. They are resistant to heat. They are resistant to detergent. They are resistant to acid and proteins. So, meaning they are more robust than the enveloped viruses. So, to destroy these viruses, you require some specific set of practice. Then, by consequence, they can survive in gastrointestinal tract, GIT. They can retain infectivity on drying. Even if it is dry, they can still be infectious. They can survive on environmental surface. And they can spread easily via format, because they don't need so must kill cell for release of mature viral particles. So they have to kill cell. Otherwise, there is no mechanism to easily get these viruses out the cell, as as there is no excretory feature component to make the cell exit from the infected host cell. Then lastly, antibody response may be sufficient to neutralize the infection, as antibodies can really close the receptor and or make this vulnerable to cell killing. They can be sufficient to neutralize the virus. So 5 basic structure of the form of virus in nature. We have NECT icosahedral. We do this, and those are example. Poliovirus is in this category. Adenovirus, hepatitis A virus, NECT helico, like mosaic virus, the tobacco mosaic virus, but no human virus is known. NECT helico, no. Enveloped icosahedral with symmetric icosahedral. We have herpes, we have yellow fever, rubella virus. Enveloped helico, so they have envelope, rabies, influenza, parainfluenza, mumps virus, measles, or paramyxoviruses. And complex, as we said, complex structure, where you don't have really clear icosahedral or helix, but these are very few, and these are mostly larger viruses like pox virus. Any question? Classification of viruses. So by naming viruses, it's more based on data available when a virus is discovered. So what is available, it can be used to name the virus, like disease that the virus is causing, like what disease. Smallpox, for example, then they call pox virus. Hepatitis viruses, immunodeficiency, human immunodeficiency viruses, HIV, measles, and they call measles viruses. Sometimes based on cytopathology occurring during infection, how the viruses modify the cell or the tissue, how the tissue appear, like respiratory syncytial virus or cytomegaloviruses. These are just named because the cell merged to make syncytial, or the giant cell, or they are just bigger, grow, and that is cytomegalovirus. Or they can also be named based on the site of infection: enterovirus, rhinovirus, adenovirus. These are basically named because of where the virus is infecting. Sometimes the place or the people who discovered this virus, like Epstein-Barr virus, Epstein-Barr was the scientist who discovered these viruses, Rift Valley virus. So the location where the virus was seen at first time. Sometimes biochemical features, retrovirus, that we said have this reverse transcriptase enzyme, and then it is retro, means reverse transcriptase viruses. However, such names are not useful for other classification. Why? Because sometimes same feature of disease can be caused by totally different viruses. So we have viral hepatitis, the virus that causes hepatitis. We have A, B, C, D, non-A, non-B, non-C, and you have others that are not yet identified. 10 to 20% of cases of presumed viral hepatitis are still not accounted for. So it's like the virus that causes hepatitis B is totally different from what causes hepatitis A. Here we have hepatinavirus. Here we have a picornavirus, and they are totally different. This is a DNA virus, while these are RNA virus. So although they cause same condition, hepatitis, they are quite totally different viruses, and they cannot then be differentiated based on the disease that they cause. The second example is herpes viruses, which is a family of viruses which cause many different diseases. We have, for example, herpes simplex viruses, which cause cold sore or type 1, or genital lesions. The type 2, the herpes simplex virus type 2, which cause a genital region. We have varicella zoster virus, which cause chicken pox, totally different from all these above. We have cytomegaloviruses, which also is herpes virus in the family, but it causes mononucleosis, a condition totally different from the above. Epstein-Barr virus causing nasopharyngeal carcinoma or Burkitt lymphoma, a condition or the cancer totally different from these, and many other herpes, human herpes virus that are not really causing any problem of this. So by having this diversity of disease with the same family of viruses, this mechanism or this way of classifying is on their symptoms, we would miss, or we cannot really find this connection between these viruses. So they are in the same family, but they cause different disease. Here we have, in previous slide, we have totally different viruses in different families, but causing almost similar or one condition, viral hepatitis. So this means classification based on disease symptoms is weak, as it doesn't allow to put together what are similar in genome or in more tangible structures of the virus, of course, although the clinical condition may be similar, like liver hepatitis, but the viruses causing that are typically different in the way they infect, the way they replicate, the genomic material are totally different. So this is a weak classification. So different viruses can cause nearly same symptom, like we said, and different members of the same group can cause different symptoms, as we said. So virologists had to use a very strict classification, which is based on features of the viruses, not on the patient or the symptom or disease caused by the viruses. So there is a classification. There is an International Committee on Taxonomy of Viruses that is used or have to meet and name viruses. And we have ODA, viral, family Viridae, Virinae, that is subfamily, virus, that is genus, then the species like tobacco mosaic, which may really connect to what this pathogen or what this virus causes, but that is fine as long as the structure or the naming is more not based on the symptom or the host. Yeah, that is fine. By 2000, more than 4,000 viruses of plant, animals, and bacteria were classified in 71 families, 9 subfamilies, and 164 genera. So these are for all, including plants, animals, and bacteria. We see that for human, what we be focusing on will not be as many as this. There will be few of them. And they seek wide range of virologists and meets every 3 years to revise the taxon or the naming, the classification. So they really do it all together with all virologists and ensure the correct naming is being so as we said, the larger or the most important character are at the top of the scale. Other characters are ranked below in order of importance until the end. We have order which have more important or most significant characters, family, subfamily, genus, strains, or types. So the classification of irrespective of their host or disease that they cause. And we have examples here of families, Poxviridae, Herpesviridae, Retroviridae, Picornaviridae. We see that, for example, the hepatitis A virus and poliovirus belong to this Picornaviridae, belong to same family, yet they cause totally different disease. So we see that they are very close to each other in terms of genetics, in terms of structure, but not causing the same disease. So primary characteristics used in classification of viruses. One is depending on their nature of the genome and their structure. So nucleic acid is the most important. When we have RNA or DNA, that is the most important criteria. They first have to do the same type, either RNA or DNA. However, among RNA or DNA, we have also a variety of conformation. We have single strand or double strand. So it may be RNA in double strand or in a single strand. We have DNA in single or double strand. It may be segmented or not segmented. That means it may be in segments. The genome is not a continuous structure, but it is segment or non-segmented. It means continuous. It may be linear or circular. So it means it can have this conformation as linear or circular. If the genome is single-stranded RNA, can it function as messenger RNA? That is where we are then classifying the two classes. Who can remind me about this? If we say, is the single-strand RNA, in case of single-strand RNA, can it function as messenger RNA? And when it functions as messenger RNA, how do we call this? When a single-strand RNA of the virus can function as messenger RNA. Anyone can explain this?
positive polarity.
That is positive polarity. So, and when it can't function as a messenger RNA, so that will be a negative polarity. Très bien. So whether the genome is diploid, such as in retrovirus, this is diploid is different from single or double-stranded. So single-stranded, double-stranded means one copy is complementary to other. While when we say diploid, like in retroviruses, you have two copies of the same configuration. It is not complementary, but you have two copies with the same configuration or with the same sequence. Do you follow? So it is different from double-stranded. Diploid means in duplicate. So these are different features that the nucleic acid, RNA, or DNA may be in the virus. And based on this, a virus can be classified together with Hello? Hello? Sorry, I think I lose my connection, but now it's okay. So the structure, the viral structure, we have seen. A question?
Yes.
Mm-hmm.
You are not getting where that point diploid and how diploid is different from double-stranded.
Hello?
Prof,
Can
you are
you
not
repeat,
getting,
please?
yes, the point about diploid and the double-stranded RNA, I mean DNA. I don't get how it is different.
Anyone can explain that? Arawamusobanurira. Arawamusobanurira difference between the diploid and the
Yes, you have said that the double-stranded DNA, it is when they are complementary sequences, two complementary sequences. While the diploid, there are two sequences, but they're similar. It's a copy of one sequences, whereas on the double-stranded, it has the complementary ones.
Very good. Is that clear to you as well? Is that clear?
No.
No?
Yes.
I show you an image. Okay. Is my image here visible to you? Not very clear. This is a representation of HIV. Muri kuyiona? A slide. Do you follow? Yeah, this is a representation of HIV virus, and it always goes with two RNA copy. And these two RNA copy are similar. They are not complementary to each other, but they are completely similar. So we have one copy here with its reverse transcriptase, always with it. And we have another copy here with reverse transcriptase. So this is diploid. So this one is completely similar to this. While double-stranded means you have a copy which have another strand complementing to this one. MRI is the complementarity of nucleic acid, where you have like a chain all close together with complementary nucleotides. The person who asked the question, is that clear? No?
Yes.
So diploid means in two copy, but same sequence. Then with symmetry, we have already seen that virion may have icosahedral, helix, or complex. It may have the outer envelope or not. It may have a different number of capsomeres, the units that make the capsid. So genetic material of viruses are such very important. In form of nucleic acid, we have single-stranded DNA. Single-stranded DNA means normally we know that DNA in nature is double-stranded, but for viruses, all are possible. We also have double-stranded as well as single-stranded DNA, which may be negative or positive. And we see that some DNA we see in replication, we require to double-strand first, and the second copy can serve as the template for messenger RNA, and that is negative polarity, while positive polarity means the messenger RNA can just be synthesized from the single-strand RNA, DNA. Double-strand DNA, we have single-strand RNA, positive and negative strands or polarity. We have double-strand RNA. We have segmented RNA configuration. So these are different types on how the genetic material or nucleic acid may be organized to the virus. And the sequence genetic organization, sequence homology, the DNA sequence, hybridization, morphology by electron microscopy, that is the outside, then the structure. some other characteristics, we will see that replication strategy is different from one virus to others, or from one family to others. Sometimes a group of viruses that seem to be a single group by the above criteria is found to contain a subgroup of viruses which have a fundamentally different replication strategy. And the replication strategy will also be part of the classification. In this case, the group will be divided based on the mode of replication. So this is the one of more important classification, which is based on the genetic material, but also how these genetic material are used to produce messenger RNA. Messenger RNA is key. You know, this is the way to protein synthesis. So with this, you already have the way to protein. So how this is produced from each type of nucleic acid, that is what David Baltimore used to classify 6 different classes of viruses. Based on 1, the genetic material